Happy to welcome everyone to this symposium titled Strategies for the Use of Real-World Data to Conduct COVID-19 Related Pharmacoepidemiology Studies. My name is Noelle Kokoris, and I'm an epidemiologist and investigator at Harvard Pilgrim Healthcare Institute, and we are the Operations Center for the US FDA's Sentinel system. Today, my colleagues and I, who I'll introduce in a moment, are going to discuss a range of COVID-19 related work that has been or is currently underway within the Sentinel system related to COVID-19. And we're going to be focusing on the data and technical issues that we've run into in these studies, the challenges, solutions we've employed, and lessons learned. The work that we're going to discuss today was funded by the US FDA and in part by the HCA Healthcare. Our views are our own, and we do not have any relevant disclosures or conflicts. So very briefly for context, I think everyone on this call knows that the pandemic has demanded rapid examination of COVID-19 from its basic epidemiology and natural history through to treatment-related assessments. As we all know, as part of ISPI, that real-world observational data have a lot of important considerations before use, and so does studying COVID-19. So coupled together, these are the major topics that we're going to cover during this panel. So we'll be talking about the use of near real-time or fresh data and the challenges that come with that and how we've approached using it. We'll talk about the need, really the underlying message is that there's a need for fit-for-purpose data assessment for every study to be done in COVID-19 where you're using real-world data. We'll talk about some of the details in terms of data variables and elements for COVID-19 in the context of lab tests, diagnoses, et cetera. And then we'll also highlight a number of collaborations that FDA has undertook, not just to promote transparency and replication of key studies, but also to really address some of the challenges that are really built into some of the real world data that we're using. And one thing to note is that much of what we'll be talking about is really in the context of the US healthcare system, specifically, particularly administrative claims data. And just to note, we're not going to be talking about vaccines today. So on the next slide, you'll see the objectives for the symposium, which I've mostly covered at this point, except to also highlight that we'll be talking about master protocols as well and how they can facilitate harmonization in terms of collaboration and study replication. So on the next slide, I will briefly introduce the rest of the panelists today. So Drs. Vin-Lo Ray of University of Pennsylvania and Dr. Wei Hua from the US FDA will be talking about the use of master protocols. Dr. Candace Fuller, who's at the Harvard Pilgrim Healthcare Institute with me, she'll be talking about the use of inpatient electronic health record data. I'll come back and talk about the use of administrative claims data. And then Dr. Sylvia Perez-Villar from the US FDA will talk about the various collaborations underway related to COVID-19. And we're saving time for discussion at the end, so please enter in the chat any questions or comments you have. So at this time, I will turn it over to Wei.
So I think Wei had some issues.
Okay, so is Wei's...
She's still working to access in. So if it's okay.
Move on to Candace.
We'll go to Candace's first. I don't think it's crucial that the order here, but I'm going to start here if that's okay, Candace.
That's perfect. I don't think the order is crucial right now. So I'm very glad to be here today. My name is Candace Fuller, and I'm a research scientist at Harvard Pilgrim Healthcare Institute. And so today what I'll do is provide an overview of some of the COVID-19 related work that utilizes inpatient EHR data in Sentinel. And so I'll focus on the challenges and lessons learned that might be useful to other researchers. Next slide, please. So epidemiologists need to understand the clinical course of COVID-19 to support assessments of safety and effectiveness of drugs. And there's a critical need for detailed inpatient data for a variety of reasons, including data lags and lack of some details that may be required by many pharmacoepidemiology studies. In the US, claims data can be inadequate for examining some of the details of clinical care received during hospitalization. So capturing and analyzing data during a public health emergency is a challenge, but the need for real-time surveillance for large and diverse patient populations is only growing. And so ongoing monitoring really helps us respond to on-demand questions, get information as quickly as possible regarding the clinical characteristics of COVID-19 patients, treatments and outcomes, and then we can adjust our understanding as more data accumulate. So when the COVID-19 pandemic began in early 2020, Sentinel happened to be working on an analysis with the FDA's Office of Counterterrorism and Emergency Preparedness, examining the feasibility of using inpatient data to examine medical countermeasures during public health emergencies. And we were using influenza as a use case. So here you see a graph where we used inpatient EHR data to examine influenza season. And this really helped us move quickly as we were able to use the data and methods we'd already developed to examine COVID-19 hospitalizations. Next slide, please. So some of the work I'm highlighting today is described in this PBS article and on the Sentinel Systems website. So feel free to take a look there if you'd like more details. Next slide. Okay, so what do we mean by inpatient EHR data? In Sentinel, some of our data partners have claims linked to EHR data. Sentinel also works with Concurrent and with EHR data aggregators such as Trinetics. HCA Healthcare is a Sentinel partner that provides frequently updated hospital EHR data. HCA captures about 4 to 5% of inpatient care in the United States, and its network currently includes 184 hospitals, which are concentrated in the southern US, especially in Texas, Florida, and Tennessee. We're working with them on what we're calling the natural history of COVID-19. And what this is an evolving descriptive analysis which examines hospitalized patients with COVID-19, and it uses standalone inpatient EHR data from 140 hospitals. Next slide, please. So inpatient EHR data can provide real-time, near real-time data for surveillance, but one really needs to think about fitness of purpose, as Noel mentioned earlier. For example, it's common to have information about high-risk conditions coded during the stay, care received in hospital, inpatient complications, but there may be very little information about care received before or after the stay. And we found it's extremely important to have a deep understanding of the source data, engage with the system, and also clinicians that are generating the data. And I'll share a few relevant examples for consideration. Next slide. So during this pandemic, there's often been a desire to have very recent data. We've discussed this a lot during this conference, but one must balance the need for fresh data with completeness and carefully think through how frequently to update the data and what data streams are required. So information we seek for a pharmacoepi study may use different data streams within the EHR. For example, we might want to use diagnoses and procedure data, as well as data from hospital laboratory systems, inpatient pharmacy administrations, or nursing documentation. And some of these will have structured fields, semi-structured fields, or totally unstructured fields, and it will differ by hospital and hospital system. So if one's working with multiple hospitals, for example, say one's using Epic, one system, and another uses Meditech, interoperability is also a consideration. So in our analysis, what we do is restrict data pools to discharged patients completely coded. This ensures we're getting complete information on the patients that are hospitalized in our analysis, but it also means we don't obtain information on patients still admitted or not completely coded. This is a trade-off, but direct engagement with system experts can really help one understand the pros and cons of different approaches. Next slide, please. So this is an example of data that was pulled in late January. If you look at the right-hand side of the slide, it looks like discharges are decreasing. But as we know, January was a time of hospitalization surges in the United States. And so data on patients still admitted or not completely coded were not yet in the data pools. When we did our next analysis, we were able to see this. But it's really important to keep these things in mind when utilizing very fresh data and figure out strategies with how you're going to handle it. Next slide, please. So how do we identify lab-confirmed COVID cases? This is an example I'm bringing up because it's a question that comes up a lot. In analysis at HCA, we access detailed information about laboratory tests conducted in the same hospital. So in recent analyses across 140 hospitals, we found 79% of hospitalized adults with a COVID-19 diagnosis code had record of a SARS-CoV-2 test during the hospitalization, and 97% of these had a positive PCR result. After we consulted with clinical and system experts, we actually learned that a lot of the patients without an inpatient laboratory test that was recorded in that system were likely tested, but some of these tests may have been prior to hospitalization or in some kind of scanned report from another facility, from a transfer, for example. And so that wasn't easily accessible to us at that time. However, we also know that the positive predictive value of inpatient COVID-19 diagnosis compared to SARS-CoV-2 tests during the pandemic is expected to be high for hospitalized patients specifically. And Noel's going to talk a little bit about some of the validation work we've done in Sentinel a little bit later on. Next slide, please. So in the US, there's a system called the Logical Observation Identifiers, Names, and Codes. It's often abbreviated as LOINC. And these are used to standardize laboratory tests in many systems. But in some hospitals, they may not use these, and they may still use homegrown naming strategies for tests, which can make it difficult to examine tests across multiple systems. So common challenges that we've seen in many different systems include that LOINC might not be available or reliable. In many systems early on the pandemic, there were codes for Middle Eastern Respiratory Syndrome, or MERS, that were used instead of SARS-CoV-2 codes, and that was just because LOINC codes for the SARS-CoV-2 tests weren't in wide use or released yet. But that's something to consider if you're using some of those historic data. You can have challenges with classifying test type with test names at different facilities. So for example, in this list, this is just an example, some of these you can clearly identify as SARS-CoV-2 PCR tests. But if you saw something like COVID-19, do you know if that's a PCR test or not? Without engaging with the hospital and the folks that work in the lab system, you may not know. So it's a consideration when using these types of data. Next slide, please. So once one figures out how to identify SARS-CoV-2 PCR tests in a particular system, one also needs to map the test results. These are just a few examples of some results, but it could be much messier. So all this is to say is really learning about the source data, cleaning and analyzing the data is doable, but it does take some time and it's important to plan for. Next slide, please. Okay, so in many inpatient EHR systems, there isn't much information about patient history beyond what is in the hospital system. And so epidemiologists need to decide what data to collect and how to balance the work required to get the data ready for analysis. So this slide displays an example analysis of some of the coded data in hospitalized patients with COVID-19 diagnosis at 140 hospitals through June 2021. And it's really just an example to demonstrate a few things. So are the coded data sufficient for your analysis? Do you need patient labs or tests? So things like smoking or obesity may not always be coded, so it could be undercaptured if you use coded data, but perhaps you may have height or weight. Is there a lot of missingness in that information? If you wanted to calculate BMI, and is that missingness differential? For smoking, maybe that is also undercoded. Maybe you want to consider anti-smoking cessation drugs, if that was something important. Malignant cancer, maybe looking at treatments might be prudent, depending on the study question. Hypertension, this uses coded data. Maybe looking at vital signs would be helpful. Diabetes, of course, there are laboratory tests. And for pregnancy, we could use ICD codes, but some hospitals actually collect this data and have a separate flag for pregnancy. And maybe that would be really a better data stream for you to use for an analysis. So a lot is possible, but it does require a deep understanding of the data, potential cleaning, and also determining what is necessary and worth effort for your particular analysis. Next slide, please. So many hospitals collect self-reported rates, and we see less unknowns in EHR data than we typically observe in claims data. This slide does not take advantage of the granularity available in race data and EHRs, but we were just trying to compare this to claims data to understand the proportion unknowns. So it was about 3% unknown in this particular analysis. However, one other consideration is the reason for missing this. So if more severe cases are missing this information, maybe there was no opportunity to collect it for some reason, it may be really important to consider an analysis. So again, knowing the data is important. Next slide. Ethnicity, as we've talked about a lot in this conference too, is a very important variable, and it's typically more complete in EHR systems as well, including in inpatient EHRs. Hispanic patients have been disproportionately affected by the pandemic, and we see that here, and it's definitely something that we need to collect, and it's very useful information in EHR systems. But there still is some unknown 4% here. Next slide. Okay, how to identify and define the exposure? Hospitals have many systems to track medication orders and administration, so epidemiologists may need to consider what is most appropriate for their analysis. We've typically used administration data, but in many systems, orders are also available, and that could be really useful for some study questions. Next slide. So in our Sentinel work, we've been monitoring inpatient medication utilization during the pandemic. And here's an example analysis, which included data through January 2021, where we confirmed that hospitalization and medication trends were generally as expected. So just a couple of highlights: dexamethasone use increased after the UK's recovery trial released their results. And we saw an increase in remdesivir after the EUA was issued in the United States. Next slide, please. So these are a few items to consider when working with inpatient EHR data in the United States, but some of these may apply to other systems and countries too. So first, determine which variables are standardized and if capture differs across hospitals. There may be no NDCs or RX norms. It may not be available in a particular system. One might need to use brand and generic names. Dose and route may not be standardized across hospitals, and so that may require some effort. Working with clinical experts to ensure complete capture of the information is really important. So for example, intraoperatively administered medications may not be captured. You may see pre and post-op, but you might not see intraoperatively administered medications. You may not know this unless you are working with some clinicians that are familiar with the systems. Multi-medication IV administered preps may not always be representative in some systems. And NDCs captured might not always represent the product manufacturer. There are very good reasons for all of these things in particular systems, but just something to consider when evaluating the data you'll be using for an analysis. Next slide. Okay, so prior to the pandemic, oxygen-related therapy wasn't a variable typically included in pharmacoepidemiology studies. But as we've talked a lot about in this conference, it's very important to COVID-19 analyses, especially when looking at severity of illness. So initially, we observed just 28% of patients with COVID-19 had evidence of oxygen-related therapies and coded data, so supplemental oxygen. And we thought that was clearly an undercapture. So our solution was, we worked with the clinicians that administered these therapies, such as high-flow nasal cannulas, to determine where this information resides. And we learned that oxygen-related nursing documentation, including devices and other notes, was where you would find this information in many systems. So we supplemented analyses with information derived from oxygen-related nursing documentation. Next slide, please. So this supplementation really improved the capture of oxygen-related therapy. So when we supplemented our analyses, 78% were on supplemental oxygen. And however, we did observe similar proportions of invasive ventilation in patients in coded data and in nursing documentation data. So that was comforting. But overall, when we look across using the nursing documentation, 79% were ventilated or on supplemental oxygen. Vast majority was supplemental oxygen, but this compared to 28% in code-based definitions. And so this example really demonstrates the importance of engaging with the clinical experts and system experts to determine where some information that you need for an analysis may reside. Next slide, please. So ordinal endpoints for COVID-19, such as the one developed by WHO, have been discussed as well extensively during this conference. And this is just an example showing what may be able to be done using inpatient endpoints when you just have a standalone inpatient EHR data. So this ranges from severe outcomes such as death in hospital to a non-ICU hospitalization without supplemental oxygen. Keep in mind, this example only uses coded data, so supplemental oxygen it may be undercaptured here, but it's just an example. Next slide, please. Here's another example showing types of analysis one can apply within inpatient EHR data to get at illness severity. So with hospitalized inpatient data, all patients were hospitalized, so we didn't have any mild cases, but 50% were moderate, 39% were hospitalized and still admitted to the ICU or mechanically ventilated in this analysis, and 11% were discharge expired. So there's a lot of different possibilities, just some examples. Next slide. So in summary, while the examples I described apply to the US, many of the considerations also apply to other systems. We found a PDS paper by Anton Podegard, which is displayed at the bottom of this slide, really helpful when we were thinking through many of these issues and considerations. And inpatient EHR data provide really important clinical information, but we found it requires a deep understanding of the underlying source data and workflows. Comprehensive identification of medications or laboratory results requires really careful mapping to comprehensively identify these things in heterogeneous systems especially. But even with direct access to data, for example, if you have inpatient diagnoses and procedures, one can miss important clinical information. Oxygen use was one example where we found it wasn't well coded, and necessitating access to nursing documentation when we were supplementing things for our analyses. We're often unable to examine patient characteristics, medication use, or care delivered before or after hospitalization unless linked to another data source. And when using data most recently pulled, it's important to implement measures to ensure completeness. For example, we use discharges for final bill patients only, and we check all data streams for completeness. Next slide, please. So finally, many individuals contributed to this work. These are just a few, and collaboration between all these organizations made this work possible. So I'll turn this over to Noelle now. Thank you.
Great, thanks, Candace. So welcome, everyone. I'm going to be talking now about using administrative claims data to study COVID-19. I'm a research scientist, epidemiologist in the Harvard Pilgrim Healthcare Institute, and we serve as the operations center for the Sentinel system. Next slide, please. So a lot of what I'll highlight will be grounded in this study. I'll use it as an example to make some of the challenges that we've run into more concrete. So this is work that Vin, Wei-Hua, myself, Vin, Sylvia, and I, and numerous colleagues at FDA and other organizations are currently leading. So we're studying the natural history of coagulopathy among people with COVID-19. Specifically, we're looking at arterial and venous thrombotic events, primarily in the inpatient setting. And we're also doing a comparison to a seasonal influenza cohort. So I'll come back to this throughout the next several slides. Next slide, please. So these are the major topics that I'm going to be talking about. So as we all know, as Candace just mentioned, EHR data have a lot of robust information, and in certain circumstances, they're absolutely the data source you need, and depending on the particular source, it may be adequate. But claims data provide a lot of longitudinality in terms of the comprehensive capture of medically attended billed care. And so depending on what you're studying, you may use claims data, or that may be all you have available. And so these are some of the considerations as you're thinking about using it for COVID-19, and some of the highlights of what we've encountered along the way over the last year and a half. So just as Candace mentioned, I will talk about data completeness and timeliness, a little bit on cohort identification and the use of laboratory test result data in these kinds of partners, challenges with identifying specifically inpatient care, as well as a little bit on mortality, generalizability, and a few other important concepts. So if you go to the next slide, please. So starting with and I'll spend the most time on this actually, on the completeness and freshness of the data. So really the question is, how fresh and complete can the claims data be that you want to work with? And really these two concepts in some ways are at odds with each other, and you have to balance the need for freshness with completeness, particularly focused on exactly what your study question is. So in the Sentinel system, our typical distributed database, which we use for many of our analyses conducted for FDA, is comprised primarily of commercial claims data, and we build in enough time to ensure that these data are complete. So typically the data are at least 6 months old, for example. However, with COVID-19, we know that we need data more fresh than that. And so we've developed the rapid distributed database for Sentinel for studying COVID-19. And it's a subset of our typical data partners. We have 2 national health plans, national insurers, and 4 integrated delivery systems. In the United States, integrated delivery systems are organizations that typically provide both health care and health insurance. And so they generally have claims data as well as some EHR data. So just keeping that in mind. And I'll talk about the concepts listed at the bottom of this slide over the next couple. So if you can move on to the next one, Vin. So this is the slide that Candace showed, and this gives you a sense of all the different data sources available in Sentinel. And depending on what FDA's question is, we use one or more of these different options. On the far left-hand side, you can see our typical claims data, distributed database with that limited network of integrated delivery systems who have access to EHR data. And we have subsetted those partners to develop this rapid COVID database. And that's what I'll be talking about And that's what we're using for the coagulopathy study. So if you go to the next slide, please. This tells you a little bit more about the rapid distributed database. These are the partners that are involved. So Humana and Aetna are the national insurers. Health partners and our 3 Kaiser Permanente colleagues at Colorado, Washington, and Northwest are participating. This is as of June. There were 55 million patient IDs in this database, and it included all patients who had enrollment from the beginning of 2020 forward, so regardless of COVID-19 status. And a couple of other things to mention about this database. So there's inclusion of SARS-CoV-2 laboratory tests, specifically nucleic acid amplification tests. I'll just refer to them as PCR. So those have been included in the data. And importantly, I'll come back to this, the national insurers do not have, or just broadly speaking, in claims data, you don't have laboratory test results, but the national insurers typically contract with some commercial laboratories in the United States to acquire laboratory test results on some of their membership. However, that data is typically limited to the outpatient setting. And so we know that there is undercapture of inpatient laboratory test results, and that'll become important in a moment. So one other thing to mention, so I'd said that the typical Sentinel data is at least 6 months old. We are having the partners actually update as frequently as every 2 months, if not more frequently. And we're still providing and conducting a really robust data quality review, the same as we do with regular Sentinel, to ensure that what we're using is appropriate for use. And we do a lot of back and forth between our partners and ourselves at the operations center. So if you go to the next slide, this is a very brief snapshot, and I won't go into detail, of the partners that were involved as of June. This is just a high-level overview of essentially the scope of this database. So you can see, for example, that there were almost 6 million COVID PCR labs as of June. There are more than 4 million COVID specific diagnoses, the U07.1 ICD-10 code. And then I'm going to talk about those 2 measures on the far right on the next 2 slides. So if you go to the next slide, please, Ben. All right, so there's a lot of information on this slide. I'll just say the high level take home, and I'm going to walk through it in detail, is that in terms of completeness and timeliness of administrative claims data in the United States, basically it depends. So depending on the partner you're working with, and then more specifically, the type of claim that you're interested in, the time to completeness will vary. The main point here is that inpatient medical claims take longer to be acquired and longer to get to completeness than other claims, such as outpatient dispensings at most of our partners. So this is showing data from one of the national insurers participating in the rapid distributed database. This is reflecting data that was generated at the very beginning of March, late February, early March. And I'll show you, I'll orient you to the graph now. So the x-axis is time, the y-axis is 2 metrics at the bottom for assessing completeness, and then above is a little bit of specificity on the claim type. And so if you look at the bottom bar, the light blue, it says absolute data partner max date. So what that means is when we look in this partner's data at this time, we see that the absolute last claim that was received was January 31st, 2021. That's pretty good for data run at the very end of February. However, that tells you nothing about completeness. So the next bar up, the purple one, the overall imputed DP max. So this is a threshold that we calculate across all of the different claims types to assess the last month when there's been an 80% threshold met that's compared to the prior month. So it's a rough estimate of completeness looking at all the different claims types. Again, not so bad, end of December. However, when you look at the specific claims types, you can see this is where things get a little bit more concerning, or at least you need to know that this is what's going on. So high level, what you can see is that for the inpatient medical claims, for data that were pulled in February at this partner, they had data through October. So you need to not only understand the overall time span of the data, but depending on what you're studying, when you're using fresh data, you have to understand the granularity by claim type. And in the coagulopathy project, we've basically looked at these metrics for every single partner every time they've refreshed their data until the time of analysis, so that we could design our study to ensure that we have adequate completeness of inpatient data, because that's our primary endpoints. So if you go to the next slide, this is just one more way of looking at this. And you can see here Again, time on the x-axis and on the y is just a simple count of inpatient encounters. This is again for one of the national insurers. And this is just a simple time series. There's nothing fancy here. And but you can see that at the end of October, the number of inpatient counters really starts to drop off. But we knew that when we calculated that 80% threshold and when we looked at that absolute maximum date, we saw dates much further out. So the point is, you need to really look at the data, speak to the partners, and understand what they have. So not only have we looked at this data for each of the different refreshes, but we've also spoken with each of the partners to ensure that what we're seeing makes sense and that we're interpreting it correctly. And so we could design, particularly the coagulopathy project, in a very precise way, but we've done this for other analyses as well in Sentinel that's used these data. So if you go to the next slide, this is just to wrap up basically. You need to balance freshness and completeness and really understand the time to completeness by both care setting and partner type. And we've built in a 4-month lag to ensure that we have good capture of inpatient claims for our endpoints of interest. So next slide, I'm now going to move to talking about cohort identification in claims data. And this is basically talking about the use of laboratory data. So as I mentioned at the beginning, the availability of laboratory test results really varies by partner and care setting, with national insurers generally having under capture of inpatient laboratory test results. And so what that means is, depending on your cohort, you may or may not want to work with such partners, or you may need other information, or you may just understand this as a limitation. And understanding the validity of diagnosis codes, as I'll talk about in a moment, might be adequate for your cohort definition. As Candace mentioned, some places use LOINCs and some don't, just as hospitals, some do and some don't, similarly with other partners for claims data. And so if you're working with LOINCs, you have to maintain those lists, make sure they're accurate and up to date. And if the partners don't have them, then it's really working with them closely to understand that their laboratory data are what you expect it to be and intend it to be. And that really gets at the last point here, which is that data quality must be reviewed and considered and really thoughtfully curated before conducting any study, and ensuring that data are mapped as expected, for example. And that's what I'll talk about on the next slide. So I think many people on this call probably know that in the Sentinel system, we have a common data model. We're using that common data model for the rapid distributed database as well. And so this is just a very small snapshot of some of the data that we're looking at. Each time the partners have updated their fresh data for the rapid distributed database. And so what you're seeing here is data from data partner 1 and data partner 2. And this is just a small amount, but high level, what you can see here is some of the information that's captured and that we reviewed for the SARS-CoV-2 tests. So you can see that the large majority are NATs or PCRs. We also look at completeness of different dates that are captured in the data. So we have both, we have all specimen collection, order date, and result date. For those that provide LOINCs, we want to make sure they're the correct ones. We've been looking at things like the percent positivity of these tests over time by partner, just to identify any anomalies or errors that may occur at the time of each refreshing. And then the distribution of patient location of these different laboratory tests. And this is really important. So as I mentioned, the national insurers will not have complete capture of inpatient labs. And even within the integrated delivery systems, you'll see a different distribution of inpatient versus outpatient laboratory tests, depending on whether or not and to what degree they own their own facilities. So that's an additional important data element to consider. So that's all I'll say for the moment on cohort identification using lab data. And now I'll talk briefly about the use of diagnosis codes for cohort identification. As Candace mentioned, I think probably many on this call know, yesterday there was a really nice session talking about validity and validation of algorithms, and comparable work was presented there. And this is actually a slide from last year's ICP, which was presented by Cheryl Kluberg, one of our colleagues at Harvard Pilgrim Healthcare Institute. And this was a validation study conducted within Sentinel to assess 5 different algorithms for identifying people with COVID-19 in the inpatient setting. So we had 5 algorithms, all ICD-10 based. You can see the U07.1 COVID-19 specific diagnosis code. That's algorithm 1. And our gold standard, so it was not a medical chart review, our gold standard was a positive PCR test. And so overall, and we also looked at these, the validity and the performance of these algorithms over multiple time periods. For the last time period available, when the analysis was conducted, the overall PPV across all of the sites for the first algorithm, which is the most simplistic of the single ICD-10 code, the PPV was 81%, which is pretty good. When we stratified by partner type, so it says their claims, what we really mean is national insurers, and integrated delivery systems, you can see that there's wide variation in the PPVs. And that's precisely because, or we think it's precisely because the national insurers simply have incomplete capture of that gold standard of a positive PCR test. We don't think it's necessarily reflective or not primarily reflective of difference in use of the code. So in the coagulopathy project, we're defining our COVID-19 cohort, our primary cohort, as either an U07.1 code in any care setting or a positive PCR test. So if you go on to the next slide, I will now just go through some of the important concepts related to using administrative claims data and wanting to assess inpatient care. So for the coagulopathy project, our primary endpoints are in the inpatient setting. They're diagnosis code based only, and we are in some ways not dealing with many of the issues listed here, at least for our endpoints. However, as I mentioned, broadly speaking, clinical laboratory test results are incomplete in pure claims sources, and so that's important to understand. If you are studying something where you need to understand the precise timing between a diagnosis, a procedure, a medication administration, for example, that's going to be really difficult, if not impossible, to see in claims data exclusively. Similarly, depending on the medication you're interested in, if you're trying to disentangle treatment and prophylaxis, for example, of anticoagulants, where you need perhaps weight or strength, dose, et cetera, you may also not be able to use claims data, and you might have to do a chart review to assess validity of your approach, or you may need to use EHR data explicitly and exclusively. And so lastly, as Candace mentioned, identifying some of the key indicators of severity and endpoints of interest for COVID-19 is challenging in general, and in claims data, it is as well. So non-invasive mechanical ventilation and oxygen use we know are undercaptured. There's generally been proxies used, and we've used these in Sentinel for ICU care, and concepts such as number of days of organ-free support is really hard, if not impossible, to capture in claims. For most of these, they're not relevant to our study, but they're really important for people to understand as you either conduct or interpret data that's from explicitly, study results explicitly from claims data. So if you go to the next slide, very briefly, again, I'm probably preaching to the choir, or at least most in this audience understand this, that mortality data in the United States in particular is very difficult to get in a timely way. In claims data, there's expected to be very good capture of in-hospital death by elements such as discharge disposition or discharge status. However, that's only as good as the completeness of your data. So again, if you know that you want to assess in-hospital death, then you need to build in whatever appropriate lag is for that data. And that's what we're doing in the coagulopathy project. Out-of-hospital death in claims data is well captured, but quite lagged. There are many sources of this information for the different partners, and really the timing and availability of that information varies by partner and partner type. We know, for example, during COVID, some states are generating their vitals information that they provide to some of the partners more frequently. And so some of the integrated delivery systems have very fresh mortality data for out of hospital. The understanding of completeness is not there though. So that's important. And then I won't talk about this in detail, but cause of death information is very challenging to collect. And there's a real delay in timeliness, and also the validity of that information is really a topic for another time. And we're not assessing cause of death information in the coagulopathy study. So lastly, there are just a couple of additional points to make when thinking about studying COVID-19 and using administrative claims data. So if you hit the next slide. So one question will be around generalizability. So the Sentinel data that we're using has a nice capture of commercially insured individuals from both national insurers as well as these integrated delivery systems. But if you really want to study something like nursing home residents, for example, administrative claims data in this country may not be the right approach given how this care is billed and captured. So really, depending on what you're studying will dictate what source you should use, of course. As has been mentioned in this conference already, the validity of the algorithms that you use is incredibly important. All of the algorithms of interest or endpoints of interest related to COVID-19 have not been validated in the ICD-10 era, including the thrombotic events that we're assessing for coagulopathy. And so we'll go back in a moment to Wei and Vin so that they can talk about the use of master protocols and how helpful that can be. And what we've done in the coagulopathy project is we took ICD-9 era validated algorithms for arterial and venous thrombotic events, mapped those to ICD-10s, and then Vin and one of our other colleagues on the project reviewed those codes carefully to ensure that they're capturing the clinical concepts. And we're actually doing a mini chart review for that project, not a full-blown validation, but really just to ensure that we've captured what we intended. On the next slide, this is a really important point, and I have a feeling this will be probably a symposium next year at ICP, which is how we'll all be impacted in our studies going forward, given the changes in care-seeking behavior and medical utilization during the pandemic. And this is really important. We're seeing this now in many different data sources in Sentinel and elsewhere in other systems I work in, where even just prevalence of certain chronic conditions, if you're looking over time, things look really funny depending on how you set up your analysis because of changes in how people sought care. And so I won't go into deep detail on this, but one thing I'll say is that for the coagulopathy project, because one of our aims is to do this comparison of the risk of the outcomes of interest to influenza, is we used a pre-pandemic influenza season as a comparison in order to avoid these changes, as well as to avoid the very low flu activity during the COVID era. And then lastly, as Candace mentioned, race and Hispanic ethnicity are important in the context of COVID-19 disparities. And depending on what you're studying, you may or may not be able to rely on claims data. Broadly speaking, these data elements are incomplete in claims data. And then the provenance of these data really varies. So some partners will have self-reported race and Hispanic ethnicity. Some will also have an imputed value. So depending on what you're studying will really impact what you use. And we are actually describing the race and ethnicity of the cohorts that we are assessing for the coagulopathy project, but we're not, for example, including these variables in any of our models because we know that they're highly incomplete in some of our partners. So in summary, these are the topics that I covered, and I think I've touched on all of them and tied them to the coagulopathy project. So on the next slide, I will just thank all of the many collaborators that have been involved in the work discussed today in terms of the rapid distributed database, the coagulopathy project, and all of our other work. Not everyone is listed here, but thank you very much. I think at this time, we're going to go back to the beginning and have Wei Hua and Vinlo Ray take over.
Okay.
Thank you, Noel. Good morning, everyone. Then I will discuss the use of a master protocol to identify COVID-19 and the relevant data in the Sentinel system. So my portion of the presentation will focus on the FDA's efforts early in the pandemic that led to the development of a master protocol. So learning from the past experience, one key strategy for pandemic preparedness and response is to build infrastructure in a timely manner. So the COVID-19 pandemic was declared in March 2020. Shortly after that, FDA formed a multidisciplinary team to establish COVID-19 cohorts for understanding of the natural history of disease and the drug safety and the effectiveness using real-world data. The team was led by the Office of Surveillance and Epidemiology and included the subject matter experts from the Office of New Drugs, Division of Antivirals, Division of Pediatrics and Maternal Health, and the Office of Pediatric Therapeutics. The work began at a point when there was a very limited data on COVID-19 disease and treatment. At that time, it was also unclear how the pandemic would evolve and how the federal and the local policies would impact the transmission dynamics. Next slide, please. To determine the scope of the cohort, we first considered the potential regulatory needs and the questions. Overall, we wanted to have a cohort that could be used to describe the course and outcomes of COVID-19 illness in various demographic groups to determine the prognostic factors of COVID-19 and the incidence and the determinants of COVID-19 complications, to study the effectiveness and the safety of COVID-19 medications, and also to provide a benchmark or serve as an external control for single-arm trials of COVID-19 medication. Next slide, please. To have a clear picture of the entire patient population, we used the concept of infectious disease transmission dynamics model and extended the model to include the subpopulation that could be observed in EHR or claims data. As illustrated in the model, patients flow from one state to another, and the parameters will be impacted by behavioral interventions and the healthcare resources. Because every component is an integral part of this dynamic system, the team reached a consensus of a cohort that is not restricted by age, sex, medical setting, pregnancy status, medication use, or any other feature. This approach would provide an integrated view of the progression of disease, capturing patient transition, and also allow for open-ended follow-up for long-term consequences. From this inclusive cohort, subcohorts could be extracted to address specific study questions. Next slide, please. To implement these ideas, we looked into clinical trials, published studies, individual case reports, professional society recommendations, guidelines, etc., to identify variables that could be potentially used to identify patients, inclusion-exclusion criteria, exposures, outcomes, risk factors, confounders, effective modifiers, or colliders in Pharmacoepi studies. We specifically considered how to align the variables with clinical trials so that the RWE findings can be compared with the trial data. One example is the baseline COVID-19 severity definition, which I will show in the next slide. In brief, to develop the list of variables interest, the team proactively explored the various sources of information in the first few months of the pandemic, and continued to update the variable list as new information became available on a daily basis. So on this slide, we show an example of considerations for alignment between RCT and RWD. The left column presents the classification of baseline COVID-19 severity as described in the FDA guidance published in May 2020. This classification is based on the clinical concepts and requires access to certain vital signs data such as respiratory rate, heart rate, and oxygen saturation, which are typically not available in claims data. The right column is a practical definition derived from the clinical concepts in the left column, but does not require vital signs data. So it represents a trade-off between accuracy and feasibility for studies using RWD. We will discuss in more detail in his presentation on this definition. Next slide, please. This table shows how we compiled a wish list of data elements prioritized for different populations of interest. Given the real-world data constraints, we did not anticipate to obtain all these variables in a single data system. The strategy was to start from an ideal situation, regardless of data source, and then consider what could be possibly implemented, and provided recommendations on how to implement in a master protocol. Now I turn over the presentation to Vin to discuss the key components of the Sentinel master protocol.
Thank you, Wei. So I hope all of you at this point are getting some sense from Wei's presentation of the value of a master protocol from the standpoint of replicability, the ability to harmonize variables and the ability to use these across the whole host of data sources, and to promote collaborations, particularly across regulatory agencies. for a whole host of different COVID-19 related pharmacoepi studies. So I think with that background, really, when we began the development of this master protocol last June 2020, again, with the goal of potentially using this for a whole host of COVID-19 related epidemiologic and pharmacoepi studies, the first question really came up with what are different approaches to identify persons with SARS coronavirus 2 infection within Sentinel particularly. And you heard from both Candace and Noel on different potential approaches. I'll give you just some of the sense that we were thinking could be utilized in the confines of this master protocol. So certainly one could approach based on diagnosis codes, you've heard some about that, laboratory tests, you've heard some about that, or some combinations. And of course, which approach you end up using really is going to depend on the type of data that you have, administrative claims versus electronic health record data. Let me first focus on some of the considerations during the development of the master protocol that we considered regarding diagnostic codes. So when we first stepped back and looked at the available diagnostic codes that might identify patients with SARS coronavirus 2, we saw these 5 ICD-10 diagnoses. And really, at the outset, you'll notice that 4 of those 5 were fairly non-specific coronavirus, non-specific SARS-related infections, and were really not necessarily being commonly used in the COVID-19 pandemic era. In contrast, the U07.1 ICD-10 code really was much more specific and was more widely being used. However, at the time, in June 2020, the validity of the U07 was really not known. Again, this ICD-10 code had just recently been established in April 1, 2020. And so as a consequence of that, the Sentinel conducted a validation study that Noel had referred to assess the accuracy of that U07.1 diagnosis code. And I think that these kind of validation studies are really fundamental to our field in order to get a sense of what is the degree of potential misclassification in using these diagnostic codes. Now, with regards to the laboratory tests, I think considerations that when we were developing this master protocol included the availability of different laboratory tests during the study period of interest, how the tests are being used by clinicians caring for persons, which are being used for active infection versus which are being used for prior exposure, and consideration for the accuracy of the test, particularly false positive, false negative rates, because this may have implications on misclassification bias. So I show in this table the 3 main forms of SARS coronavirus 2 laboratory tests: the nucleic acid amplification tests, the rapid antigen tests, and the antibody test. And I think it is important to consider each of the aspects of these tests when making a decision should be used in your own pharmacoepidemiologic analyses. And I'll give you just some of the considerations that we provided within the master protocol for investigators to consider. Certainly the nucleic acid tests, they are the most widely used tests. We're talking, these are the RT-PCR tests. They have high sensitivity, have high specificity. The antigen tests, while rapid, have somewhat high false negative rates, and particularly high on the upwards of 20 to 30% false positive rates, even asymptomatic, which makes these somewhat challenging to potentially use within pharmacoepi studies because of potential for misclassification. And of course, antibody tests are really not being used clinically to identify active infections, but are really being used to determine previous viral exposure or immune status. And so, one of the things that we suggested in our master protocol is that really the focus should be on nucleic acid tests, just given all of the error that is associated with the rapid antigen test. Of course, those are decisions that are going to need to be made differently in different locales, different geographic locations with different tests. But this was the issues inherent in the tests in the US. So we provided different approaches to identifying persons with SARS coronavirus 2 infection in the Sentinel Master Protocol to allow a sort of menu of options that investigators going forward could consider, depending on the type of electronic data sources that they had and the types of studies that they were interested in conducting in the future. So, we suggested that potentially, if you had access to both claims and electronic medical records, you could potentially implement algorithms that included both ambulatory or hospital discharge ICD-10 UO7.1 diagnosis in any position, or a positive SARS nucleic acid amplification test. Really implementing this on or after April 1, which was the date that the ICD-10 UO7.1 became available. And for the standpoint of laboratory tests, utilizing the specimen collection date as, in essence, the index date for the laboratory test. We also suggested that potentially doing either sub-analyses or exclusive analyses among individuals who had positive nucleic acid tests from any location would be viable, and to consider index diagnoses in hospital settings and in ambulatory settings, as these may reflect different populations with different risk factors. Now, Wei had alluded to analyses of the severity of COVID-19 in part of her talk, and others have mentioned that this is an important covariate, and as a consequence of that, when we developed the master protocol, we spent a lot of time considering and deliberating what should be the variables that are included and what should be the stages of severity of COVID-19. And we ended up coming up with two schema. This is the more complex schema that had several levels, a number of levels of COVID severity, and you could see at right each of the criteria. And the criteria here included symptoms such as fever, cough, sore throat, non-specific symptoms that might be related to COVID, as well as pneumonia diagnoses, use of therapies like oxygen, ICU admission, organ failure, mechanical ventilation. And we recognize that this is going to be a complex algorithm to implement to classify COVID severity. Symptoms are not typically recorded in administrative data. We recognize that if investigators wanted to utilize this sort of scheme, they would need to go back to medical records using things either like natural language processing to extract symptoms from records, or go into medical records themselves and extract from progress notes. And so, recognizing that this might be more complex to implement, we also included an alternative, more simplified schema, but putting on my own hat as an infectious disease clinician, which I thought was actually still quite relevant, where you have somebody who is classified as mild infection at the index diagnosis, they've not been hospitalized. Somebody with moderate infection, they have been a moderate COVID-19 severity, they have been hospitalized, but yet they've not been admitted to either an ICU or mechanically ventilated, recognizing that sometimes with ICUs at capacity, patients may not necessarily be cared for in ICU settings. They may be in sort of MASH hospitals or trailers, but still receiving ICU level care, maybe even mechanically ventilated. So we wanted to have some freedom with that. And then a third, more severe COVID-19 severity, classified by you're hospitalized and you either got ICU level care or you're mechanically ventilated. And this was a schema that we thought in more general pharmacoepi analyses in the claims or EHR data would be applicable and potentially more feasibly implemented. Now, we spent a lot of time, and Wei alluded to, about the variables that we wanted to collect. And certainly we started out with a more aspirational list, but we classify what we really thought were critical to collect as data elements in the baseline as well as in the longitudinal data. And we felt that from the baseline period, from whatever baseline data in some period prior to your COVID-19 diagnosis, certainly demographic data, pre-existing conditions. I mean, we've heard a lot in the literature about the importance of pre-existing cardiovascular disease conditions, on COVID-19 diagnosis and COVID severity, pre-existing medications that might have implications on COVID complications, laboratory results, vital signs, radiographic reports, all may be of relevance depending on the type of question you're going to answer in your COVID-19 related pharmacoepi study. From the standpoint of longitudinal data that investigators might want to collect, what we included in the master protocol was not only medications, labs, vital signs, and radiographic reports, but also certainly a whole host of SARS treatments, COVID-19 supportive therapies, and recognizing that over time we may need to update these as new data, new therapies were being implemented for the care of patients with COVID-19. And at least at the time when we developed the master protocol, we came up with a list of what we thought were relevant COVID-19 related complications. And you certainly heard both Noel and Candace allude to some of these arterial and venous thrombotic events, certainly pneumonias, bacterial infections, pregnancy outcomes, and all-cause mortality. And then, given the complexity surrounding these variables within the master protocol, we spent a fair amount of time considering important aspects, such as the definitions for the duration of baseline period, which may need to be different depending on whether you have electronic medical record data, in which case maybe you'll want to have a longer baseline period versus a claims-based baseline period. The need to explore the ascertainment and completeness of variables for which within Sentinel, we may not have necessarily really used or understood the ascertainment and completeness. And so deeper dives would be necessary for things like SARS laboratory tests, D-dimer for the analyses of arterial and venous thrombotic events, oxygen saturations, and inpatient medications. Paying attention and considering missing data. You heard from Noel about race, symptoms, over-the-counter medications, as well as the uncertain validity of variables like supplemental oxygen, like the COVID-19 severity schemas, in which reviewing medical records, performing validations would be valuable. And then we concluded this master protocol by really thinking deeply about the potential limitations that may be inherent in pharmacoepi analyses related to the data and the study designs, and formulated ways to potentially address or mitigate these limitations. So we talked about things like misclassification bias, particularly with regards to SARS diagnosis, either by diagnostic codes or labs, COVID-19 severity, the use of supplemental oxygen, and Candace went through a lot about the deeper dives related to supplemental oxygen and what kinds of data. And we recommended that depending on the different data sources and what had been previously done, additional validations may be necessary. We talked about selection bias and the fact that there may be variability in SARS coronavirus 2 lab testing over time, by disease severity, and by geographic location, and recommended to investigators sensitivity analyses that might be valuable according to time periods, COVID severity, the setting of diagnosis, ambulatory, hospital, and geographic locations. We recognize that investigators who might be interested in medication exposure and its impact on COVID, potentially infection and other COVID outcomes, protopathic bias would be of important to consider, where medications may be prescribed for early symptoms of disease in those who have not yet been diagnosed. I'm thinking about drugs such as nonsteroidal anti-inflammatory drugs. And so we suggested that potentially sensitivity analyses to examine different exposure windows prior to COVID-19 diagnosis would be sensible. And then lastly, you heard from Wei, she alluded to different colliders, and we recognize that collider bias may be at play in COVID-19 related pharmacoepi studies, whereby the likelihood of being sampled may relate to different risk factors and outcomes of interest, such as age and obesity colliding for COVID-19 testing, or smoking and COVID infection itself colliding on hospitalizations. And we suggested potential sensitivity analyses implementing probability weighting of sampling or negative control analyses. So in summary, I hope hope Wei and I have given you some sense of the value of this master protocol that Sentinel developed early on in summer 2020, given the global need for rapid acquisition of data across different settings for different subgroups. These master protocols can really facilitate harmonization of study designs and methods across different data sources. They can facilitate assessment of the homogeneity and heterogeneity of findings across databases, across regions, across countries. And I think, not to be understated here, these protocols could be utilized by other different regulatory agencies to promote collaboration, to promote communication, which I think, in this era of COVID-19, we all heard about misinformation, disinformation, and infodemic is incredibly valuable. So I put at the bottom of the slide the website where this master protocol is available, and I just want to acknowledge my colleagues in Harvard Pilgrim Healthcare Institute, FDA, our funding source, without which this work couldn't have been done. Thanks for your attention. And I'm going to turn this over now to Sylvia Perez Villar, who is going to talk to us now about how collaborations with researchers using these master protocols have really facilitated COVID-19 related pharmacoepi research. So Sylvia, turn to you.
Thanks, Min. My name is Sylvia Perez Villar. I'm a senior epidemiologist in the Office of Surveillance and Epidemiology in CDER FDA. We are going to discuss now how the FDA has promoted collaborations with researchers, data providers, and regulatory agencies in the time of COVID. Next, please. For that, we are going to provide some examples of these collaborations initiated either by the Office of Surveillance and Epidemiology and by the Office of the Commissioner. Next, please. First of all, why these collaborations? Because of the need to enhance validity and generalizability, because we needed to increase sample size of certain subgroups of interest, such as frail, elderly, pregnant women, immunocompromised individuals, because availability of additional data elements, such as inpatient medications, race, more granular data to characterize disease severity, because of the need to look for consistency of results despite differences in data systems and environments, and to promote national and international collaboration in the middle of a global pandemic. Next, please. Before we go to the examples, let's provide some context. Since the pandemic started, leadership and the COVID-19 scientific activities core team in the Office of Surveillance and Epidemiology have been overseeing and coordinating pharmacoepidemiologic efforts to timely address relevant questions, safety concerns, using Sentinel data and interagency agreements with other federal agencies, including CMS and the US Department of Veteran Affairs. Next, please. The Office of the Commissioner is also leading efforts to use real-world data to advance the understanding of the natural history of COVID-19 specific patient populations, as well as treatment and diagnostic patterns, and to develop methods to streamline clinical and post-marketing evaluation of FDA-regulated products. One of the efforts I would like to highlight here is the assembling of an interagency and multidisciplinary team, which is directly working with data vendors through research collaboration agreements and short-term contracts. Next, please. Another effort promoted by the Office of the Commissioner has been the COVID-19 Evidence Accelerator, in which the FDA and the Regal Udall Foundation work closely to develop key research questions that multiple researchers and data providers have been addressing through parallel analysis. After the questions were set and the protocols provided, the accelerator revised together a list of core data elements needed, identified critical elements to answer a research question, established uniform collection parameters, discuss details on the implementation in their databases, shared a list of codes, and provided feedback to other teams. Next, please. Finally, another key element has been the International Coalition of Medicine Regulatory Authorities, in which international regulators agreed to cooperate in building international clinical cohorts of COVID-19 patients to conduct research using real-world evidence, share expertise, and increase study power and data quality. Next, please. Let's discuss now some examples of the results of these collaborations. Next, please. Because of the public health need to provide emergency use authorization for COVID-19 treatments that meet legal criteria, the Office of Surveillance and Epidemiology developed an active safety surveillance plan and decided to use the surveillance of monoclonal antibodies authorized under emergency use authorization as the use case. Next, please. This study aimed to conduct monitoring of the uptake of monoclonal antibodies under emergency use authorization, and depending on the uptake, to conduct monitoring of pre-specified safety signals, and in case a signal is detected, to also conduct signal refinement and evaluation analysis. Next, please. This surveillance is currently being conducted simultaneously in the Sentinel Rapid COVID, CMS, and the VA databases. We used a common protocol, which, as in the rest of examples we are going to see today, had to be adapted to fit the data characteristics of each system, while we try to maintain consistency across the different analyses. Next slide, please. In this particular example, we are using FDA's severity definition and the master protocol, described by Wei and Dean just now. By implementing the study in these three data systems, Sentinel, Rapid COVID, CMS, and VA, we are able to monitor monoclonal antibody use in large populations, having a high representation of commercially insured individuals, ages 18 to 64, a large elderly population, and also a population with a largely male, more racially diverse, and relatively sicker population for their age. There are also other differences between systems that we are also able to capture. For example, in the VA, prescribers are required to fill out a form to make sure that the patient is eligible for monoclonal antibodies according to the emergency use criteria, which does not happen in CMS and Sentinel. From these, if you can go back a little, yeah, thank you. From these collaborations, we expect results that should help inform regulatory decision-making and development of methods with application to other emergency use authorization. We also expect that this work will help us refine the applicability of the FDA's severity definition in both claims and EHR data systems. Now, yeah, go back to the next one. Another example is FDA's monitoring of corticosteroid use in non-hospitalized patients. Next, please. In this study, we aim to examine utilization patterns, characteristics, and outcomes among COVID-19 patients with evidence of systemic corticosteroid initiation in US outpatient settings. Next, please. Again, using a common protocol that again had to be adapted to fit the data characteristics of each system, while we try to maintain consistency across the different analyses. We conducted this descriptive analysis in Sentinel Rapid COVID, CMS, and the VA. For this particular example, we also use health variety data through a research collaboration agreement established by the Office of the Commissioner. Next, please. As part of these collaborations, we were able to examine clinical and demographic characteristics and patterns of use of systemic corticosteroids in COVID-19 outpatients using data from 4 large databases representing most of the US, large populations of COVID-19 outpatients, and diverse representation of all ages. The results have been presented to the NIH COVID-19 treatment guidelines panel and to the International Coalition of Medicine Regulatory Authorities, and the common protocol and the Sentinel SAS code have been shared with Health Canada's contractors. Next slide, please. Noel discussed at the beginning of the presentation the challenge we encountered with the implementation of the study and natural history of coagulopathy in Sentinel. We are going to use now this same example to also show how we use this study to work with researchers, data providers, and international agencies. Next, please. This study, which is led by Dean, aims to assess incidence and risk factors for arterial and venous thrombotic events in COVID-19 patients and compare the risk with that of individuals diagnosed with influenza. Next, please. Besides in Sentinel, this protocol, or parts of it, have been implemented in several databases through the collaboration with the Regal-Udan Foundation, has been identified as one of the projects for the International Coalition of Medicine Regulatory Authorities, and adapted for implementation in databases across Europe and Canada. Once all these studies are completed, which should be soon, we expect to have robust national and international data to better quantify the frequency and consequences of COVID-19 related thrombotic events, to better identify subgroups at high risk for these events, and also help inform benefit risk assessments. Next, please. This COVID-19 pregnancy study is another example that also illustrates the benefit of an international collaboration. In this case, FDA used EMA's protocol to inform the development of its own protocol. Next, please. This study, led by Wei, aims to assess drug use and describe severity and clinical outcomes in pregnant women with COVID-19 according to treatments received during pregnancy, and compare this data with those of non-pregnant women with COVID-19. These are two of the three study aims in the EMA-funded study. Because as I mentioned, please, next slide, please. Because as mentioned, this protocol, which is currently being implemented in Sentinel, is based on the EMA-funded Consign, a project being implemented in a variety of data sources across 8 European countries. To design this, to adapt the protocol to the US setting in Sentinel, FDA has also used the FDA's master protocol in its design. Next, please. These two examples, natural history of coagulopathy and the pregnancy study, can help us learn about natural history, patient characteristics, treatment patterns, outcomes associated with management strategies for COVID-19 in different health systems and environments. Also help us rapidly assess consistency of findings, enhance validity and generalizability, increase sample size of subgroups of interest, and because of all that, to improve efficiency of regulatory processes and decision making. Next, please. Therefore, in summary, this pandemic has shown us the value of strengthening of existing systems already in place prior to the pandemic, timely collaborations within and across agencies, transparency and information sharing, coordination leading to parallel and joint analysis, and validations of databases for future use. Finally, I would like to thank all our colleagues and collaborators from FDA, Sentinel Operations Center, Penn, Aetna, Humana, Kaiser Permanente Washington, Kaiser Permanente Colorado, Kaiser Permanente Northwest, Health Partners, the VA, CMS, Acumen, Aetion, FDA contractors with the Office of the Commissioner, the Regal Udall Foundation and Evidence Accelerators, the International Coalition of Medicine Regulatory Authorities, EMA, Health Canada, and their contractors. Thank you.
Terrific. Great. Okay, I am going to stop sharing. So we have some time for questions. And I would like to welcome any of the questions that our audience has. Please feel free to enter your questions either in the chat box or you can you raise your hand. I'll do my best to try to look through, but it may be more efficient if you put questions in the chat box. We have a couple of questions so far. I know that Candace has answered some of these. Looks like Wei has also. I think there's a question about long COVID and how might Sentinel record and categorize complications related to long COVID syndrome for pharmacoepi analyses. I don't know if.
So I know that this is Noelle. Wei responded in the context of the master protocol. I think the other thing I can add is, as far as I'm aware, there's no currently validated or developed or validated algorithm for long COVID in the US, at least based on my last review somewhat recently. And there is a WHO code for it, but that's not been implemented in the US as far as I'm aware. So I think there's a lot to be done there, and it seems like it's being defined differently in different studies based on the ones that I've seen at least. And at this time, we're not currently studying long COVID within Sentinel, but not sure if others want to add anything to that.
Yeah, I mean, I would echo that. I think first and foremost, I think from an epidemiological standpoint, you'd have to come up with a phenotype, and that phenotype, we'd have to have some kind of validation for that phenotype, which would make it challenging at this point to study, I think, long COVID without any kind of either ICD-10 diagnosis or phenotype would make it challenging in the confines of these kinds of studies. Studying long COVID might be more amenable in a sort of prospective cohort where you're administering questionnaires to patients, over time to assess symptoms and outcomes. But to follow up on what you're saying, Noelle, I think that it's rich, fertile area for research, both from the standpoint of, what kinds of medications might either mitigate or exacerbate long COVID symptoms, or, long-term natural history and outcomes. Looks like Danny Prieto Alhambra also said, not used in the UK either, I'm afraid at this point. So another question, I think, let's see, Candace has tried to, well, I guess just to follow up, somebody asked Elizabeth Monsi, is there an ICD diagnosis code for history of clinical COVID-19? Anybody want to, I know Candace or?
Yeah, there's as I understand, I don't think it's been implemented in the US. I think this is another one of those. Candace might know better, but I know that there's one from WHO, but I don't think it's implemented here yet. Is that right? Or has it gone into effect?
Now, there's some other codes that you could use for a history of COVID, but you don't know for sure if that is long COVID syndrome and what that actually means. So certainly with codes, I think it's a challenge. It sort of reminds me when the COVID pandemic began, we didn't have a specific code for COVID at that time either. So we were using all kinds of codes and trying to describe the symptoms and figure out a phenotype. I think there are a lot of researchers doing that right now to try to understand to define it and figure out how it would be captured. So I think that will be a big thing this next year. A lot of people will be doing work on this.
So I know that there's a lot of questions about breakthrough infections after vaccination, and there's a question from I think it was Sylvia Taylor. We didn't spend a lot of time talking about COVID-19 vaccination status. We just had certainly a plenary about vaccines, but there is a question here. If a study is not aiming to assess vaccine effectiveness or safety, nevertheless, knowing a patient's vaccination status can be important. Do you think such data may be better recorded for some special subpopulations, cancer, immunocompromised patients, and claims EHR? What are some of the challenges or nuances in which we have worked to assess, if at all, COVID-19 vaccination status?
I can tackle this a little bit. This is an active area of interest. We know how important it is to assess vaccination status, and we're actively working on that to try to understand. So in a lot of inpatient EHR systems, it might be captured, it might be self-report. And so who is reporting this? So it is a challenge, and I think we'll have more information over time. I believe some of the other sessions have described some of the challenges we've had with capturing vaccination, using claims data and other sources, but clearly it is something we need to improve upon, and I think we'll be working on this as well next year.
Yeah, I mean, to the question that was asked, it may very well be that capture of vaccination status may actually differ in these data sources depending on underlying comorbidities like immunocompromised states, for example. But I think, with so many people going to local pharmacies, out of network, potentially out of network settings to obtain COVID vaccination status, it is a bit of a challenge to classify that. Can you speak to plans for publishing validation studies? This is from Courtney Davis. Can you speak to plans for publishing validation studies as they become available over time, since these can impact other research protocols to improve quality? I guess, let me ask this to you, Noel, because you had brought up the validation of the U0701, and then maybe also ask Candace, because the validation about supplemental oxygen use might also be of interest. Just maybe want to comment on plans for publications, and then unfortunately our time is up. We'll have to close there.
Yeah, the validation study I mentioned is under review at a journal now, and actually it was presented at last year's ICPE. So I'm sure I can share the slides if you'd like, but it hopefully will be published soon, at least that one.
And Candace, anything from your staff?
Sure. So we do have some preliminary results on the oxygen analysis in our PDS article, which is in this slide deck, but I think it may be worth writing up a little bit more detail. And we always post all of our papers and analyses on the Sentinel website too, so certainly monitor that.
I mean, I think any publication of these are going to be useful. Of course, the transportability of any of these algorithms is going to be, certainly of question whether or not, that would be valid in different data sources, may warrant separate validation studies is a separate issue. Well, it is 11:31. It's the end of this session. I want to thank all of our panelists for the excellent discussions. I want to thank all of our virtual audience for their participation in this session. As I said at the outset, the slides and the recording will be available on ISPE's website, and hope this was valuable for all of you. And thanks so much as we continue to try to advance the science in this area. Best wishes all.